Hi, everyone. My name is Emma Bollig, and I have the privilege of introducing our first speaker. Dr. Granick is an associate professor in the Veterinary Clinical Sciences Department and a member of the Small Animal Internal Medicine faculty. She leads the VMC's Antimicrobial Stewardship Committee, is a founder of the Antimicrobial Resistance and Stewardship Initiative at the University of Minnesota, the AHA representative to the AVMA Committee on Antimicrobials, a co-chair of the World Small Animal Veterinary Association's Therapeutics Committee, and a founding member of the new Companion Animal Antimicrobial Stewards Network that unites those working in veterinary academics, regulatory agencies, and industry to make progress in antimicrobial stewardship.
Thanks, Emma. And then for those that are on Zoom and didn't hear before, Emma is my partner in crime. So basically everything I do is possible because she is working with me. So today I'm going to talk about antimicrobial use studies. We collect antimicrobial use data from dogs and cats in veterinary clinical practice. And the drive behind that is not just because it's fun to do, but because we think that data is meaningful in terms of moving the dial on the way that we use and optimize use of antimicrobials. So I'm going to talk really briefly about why we collect this data and then provide some highlights from a few of the studies that we've done, so not getting too in depth. Two of the three studies that I will highlight today have been published. So if you're interested in the details, you can look at those publications, and one hopefully shortly will be published. We're just doing some revisions. So, and again, I will share with you as we go through each of them sort of the insights that the data provides to us in terms of how do we improve antimicrobial use. So we all know antibiotics are really important for animal health as well as for human health. But the truth of the matter is that all antimicrobial use leads to the potential for antimicrobial resistance. Bacteria are born to survive, right? And so they continually find new ways to subvert the effects of antimicrobials. And so what that means for prescribers, veterinarians in this context, is that we need to use these drugs judiciously in order to slow the development of antimicrobial resistance and keep these drugs efficacious for our clinical purposes. So in an effort to improve antimicrobial use, the American Veterinary Medical Association has created the core principles of antimicrobial stewardship in the context of veterinary medicine. And there are lots of details. If you're interested, I'll talk to you about it ad nauseum at some other point. But one of those core principles is to evaluate antimicrobial drug use practices. And so that's what we're going to be focused on today. And if you look at these core principles, AVMA gives us some more details. So what they say in terms of evaluating drug use practices is to encourage development of programs for the evaluation of antimicrobial drug prescribing at both the veterinary practice and the aggregated levels. And I'll show you examples of both today. If we're going to collect this data, we need to ensure that this data gets back to veterinarians, because otherwise, what's the point? And then they also encourage supporting, analyzing, and sharing of drug use data, all the while preserving that veterinarian client confidentiality. And so all of the projects we do, the point prevalence surveys that I'll talk to you about today, as well as a larger project where we collect medical record data from dogs and cats across U.S. veterinary practices, are one of the key elements there is to keep that data anonymous and confidential. So I've alluded to this already, but what's the point of tracking antimicrobial use in dogs and cats? Well, we can use the data that we collect to establish baseline measures to understand what we're doing, because we don't know where we're going to go if we don't know where we are right now. That data can provide targets for us to set goals in terms of changing the way we use antimicrobials, changing the antimicrobials that we use, and also goals for providing education so that those things can happen. And then if we can provide appropriate interventions and education and other support, then we can repeat these measures over time to see if we're making progress or if there are new issues that are arising. So basically, you can't manage what you don't measure. And so this is an effort to measure what we're doing. And this is not something unique to veterinary medicine. In fact, in human healthcare, antimicrobial reporting is required, and that data gets sent to the CDC. CDC uses some of the methodologies that I'll talk about today to collect data that they don't otherwise get from electronic health records. So the methods that I'll be talking about today is the point prevalence survey method. So this methodology is with the idea that collecting antimicrobial use data doesn't have to be super burdensome. If we can collect data in a uniform fashion across multiple institutions, then it's not a lot of lift for each institution. But if we collect that in a uniform standard manner, we can aggregate that data and say something bigger. And so point prevalence survey methodology typically is collecting data from a single day of practice. And across multiple sites, we can collect data prospectively. All of the data I'll share with you is collected retrospectively. There's lots of reasons for that, but one is that it takes veterinarians a few days usually to complete their medical records. We can repeat this methodology over time, and we can use it to estimate both overall prevalence of antimicrobial use, and then also, if you collect the right data, appropriateness of that use for specific clinical diseases. We collect data using the REDCap tool. Actually, the first study I'll show you, we used Excel, and then we quickly graduated from that. And the key here is that we created really specific and defined SOPs for data entry and data dictionaries that defined every single data element that we collected to make sure that there was standard collection of that data across all of the institutions that we collect. We've done a bunch of these and we've learned from each one of them. So I think we're actually pretty good at it now. The first time that we attempted this, we did it just within our VMC, so our veterinary medical teaching hospital here. We collected data 1 day a month. Because we were only collecting from one institution, we had to collect from multiple days. And we finished that up in 2019. In 2020, we collected data from 19 practices, companion animal and mixed animal practices in Minnesota and North Dakota. And we did that quarterly because we only had 19 practices. And some of them, were small and some of them were bigger. But again, to get a bigger N that we could say something about. In 2020, we did our first US teaching hospital point prevalence survey. We had 14 hospitals that participated, which I would say is not bad, given that, if you all remember, that was COVID year. And we had, we repeated that in 2022. We expanded it to Canadian teaching hospitals as well. And we had, oh, sorry, that's not 52. Emma, how many did you have?
31.
26
US, 5.
26 US, and then 31 total practices. In between the two veterinary teaching hospital studies, we did a non-academic hospital. So that included referral hospitals and primary care practices, and I'll share some of that data with you. And we had 52 of these hospitals participate. And then we kind of switched it up because we wanted to know a little bit more about certain diseases, especially those that weren't well represented in our data sets. And so we kind of switched a little away from the point prevalence survey, but used a lot of the same methodology. And we focused on dental referral practices in the US as well as dermatology practices. And instead of collecting data about 1 day, we created a threshold of the number of patients that we wanted them to collect data about. And then all of these studies starting in 2020 were at least partially or fully funded by the FDA. And we have a cooperative agreement with them to collect antimicrobial use data. So first, I'm going to share with you the study that we did here in the teaching hospital that went between 2018 and 2019. We collected data on the first Monday of the month. We did a little data analysis ahead of time to pick the best day of the week that would be most representative. We collected data from our electronic medical record, laboratory reports, and treatment sheets. And our goal here was to define the prevalence of prescribing in our teaching hospital and describes the drugs that were used, the classes, and the indications for use. And so these are the specifics of the data that we collected, and I won't get too in the weeds there. I'll just kind of give you the highlights. So in that 1 day a month over 12 months, we collected data about 856 patients. 25% of those were inpatients and the rest were outpatients. Most of those animals were dogs, 72%, and that sort of balance of dogs and cats has held up pretty consistently over all of the studies that we've done. There are more cats in the US than dogs, but they don't go to the vet. And then there were lots of interesting findings here, but one of the things that struck us was that we had quite a lot of prescribing of nitroimidazoles, and the primary drug here was a drug called metronidazole, and that's an antibiotic that, in my opinion, has like very little actual like real clinical appropriate use. And when we looked at what we used metronidazole for, it was for cases almost exclusively, not totally, of acute diarrhea. And acute diarrhea is a really common disease process in dogs. In fact, if you look at the numbers of GI disease across all of our point prevalence surveys, we're in that like about 17% of outpatients have GI disease. And so our outpatient prescriptions, almost a fifth of them were for acute diarrhea and they were metronidazole prescriptions. And so we decided that this was a key area of antimicrobial stewardship. And the time that we did this study, we were just starting out having an antimicrobial stewardship committee in our veterinary teaching hospital. And so that was one focus that we wanted to try to make some change, get some traction for change in the hospital. 39% of our prescriptions were also given to patients that had no recorded evidence of infection. And there are lots, if you're a veterinarian, you know, there are certainly situations where we can't get, a culture or a sample to prove an infection, and we are basing our treatment on our high index of suspicion, but it is an area where we could probably do better in general as a profession. So what are the things that we could do? Well, we can provide guidance for alternative treatments for acute nonspecific diarrhea, and those include providing prescription diets with prebiotics that support the gut, probiotics that also support the gut, and then educating folks about the risk of use of metronidazole. And I won't get into all of that data, but there's a lot of data and new guidelines out of Europe that suggest that we don't need to give antibiotics to these patients, and the use of metronidazole can actually be detrimental. And then we want to, focus on prescribing, trying to really think critically about when we're prescribing for patients with no evidence of infection, because what veterinarians like to do when they don't have diagnostics to support them is to prescribe antibiotics just in case. So that's a really hard dial to move, but that's something that, as Emma mentioned, we have a group now networked across the US that's trying to make changes there. So this is just an example of what the outcome of collecting such data can be in the context of our veterinary teaching hospital. So we continue to monitor metronidazole prescribing over time. And then we had a bunch of different interventions starting in 2018 to the present. And those included things like creation of our informal antimicrobial stewardship committee that supported people's prescribing decisions. We created a pocket guide for antimicrobial prescribing that suggested not prescribing antibiotics for diarrhea. We later formalized the antimicrobial stewardship committee in the hospital and became more available for consults. We made sure that we had dietary alternatives as well as probiotics in the places that veterinarians needed them, like in the after hours pharmacy, so that, at night and on the weekends, when those diarrhea cases were coming in, that the veterinarians that were prescribing metronidazole had an alternative. And then we provided some educational opportunities in the form of newsletters and other things, grand rounds and so on and so forth. And you can see that was a pretty effective intervention. And I think the key was multiple interventions over time. So now I'm going to switch gears to, there's a lot more in that study, but I think that was kind of one of the highlights that came out of it. As I mentioned, we did a point prevalence survey of antimicrobial use in 52 non-academic hospitals in the US. They selected one day in a 2-week period that was representative of their regular daily practice in August of 2021. There was all sorts of data that we asked folks to collect. They collected data about animals that received antibiotics and those that did not, so we could get a prevalence of antibiotics. And then we also asked information about what the disease process that they were treating, whether there was evidence of infection, was the intended reason for treatment, was it to treat infection, was it prophylactic treatment, was it some other reason, and then we gathered information about the antibiotics that were prescribed. So we had pretty good representation in all of the major areas in the United States, so Northeast, South, Midwest, and West. And in total, we had almost 2,600 dogs and cats that were included. Again, about 3/4 were dogs and 1/4 were cats. We had 32 primary care practices and 19 referral practices that participated, and we had a single animal shelter. And most of the patients that were included in this study were outpatients. 62% of all prescribing in these practices was for the treatment of infection. Not all of that infection was confirmed. So of those, only 31% of cases had confirmed infection. You can see the vast majority of cases were just suspected. And we had pretty strict definitions of what each of those things meant. For prophylactic purposes, which was a little over 1/4 of all prescribing in these practices, cefazolin was the most often used antimicrobial, but you can see that cefpodoxime, which is a 3rd-generation cephalosporin, was also used for prophylaxis and probably not a recommended first-line treatment for that purpose. And you can see here for outpatient treatment, look, metronidazole shows up again, 14% of all outpatient prescriptions. So we were able to identify what the most prescribed drugs were and most common antimicrobial prescribed in dogs and cats. And this is true if you look at antimicrobial use studies in other settings too, like in Europe, in England, in Australia, where they've been looking at prescribing in dogs and cats. Aminopenicillins with beta-lactamase inhibitors are very commonly used, the #1 drug. And then the #2 drug is those imidazoles, the metronidazole, and then we move on from there to cephalosporins. Most of the time that's prophylactic use, and then 3rd-generation cephalosporins also show up quite commonly as well. Why is this important? It's important for our veterinary practice, but all of these drugs are used in human healthcare as well. Some of them are higher priority drugs, including the fluoroquinolones and the 3rd-generation cephalosporins. In the EU, there are now restrictions to prescribing those for veterinarians, and in the US, we can do whatever we want. So another reason to try to get these numbers in front of veterinarians. When we looked at differences in prescribing between primary care practices and referral practices prescribed more overall, but primary care practices prescribed more 3rd-generation cephalosporins than referral hospitals. And that's primarily driven by a drug called cefovecin. It's an injectable 3rd-generation cephalosporin. Veterinarians like to use it for cats because cats are not easy to pill. The problem is, 1, it's not a first-line agent. And then 2, it lasts for 10 to 14 days. So the duration of treatment using that drug is often longer than we need to use for most things. So when we look at duration of therapy, if we look at prophylaxis versus the intent to treat infection, you can see, just ignore everything else. And some of drugs are excluded because they just didn't fit on here. But what's in yellow is the most important thing, which is that for prophylaxis, the prevention of infection that may or may not occur, we're treating for, on average, 10 days, right? Actually, that's a median, but and then when we're treating infection, 10 days. So it doesn't make a lot of clinical sense that we are treating as long to prevent infection as we are to treat actual true infections that are there, or at least we suspect are there. And so this is an area of antimicrobial stewardship effort in human healthcare, and it's becoming increasingly focused on in veterinary healthcare too. So it's great that we have this baseline data, and what we hope to see is that as we continue to do these studies, that this number for prophylaxis will go way down. In fact, there are surgical prophylaxis guidelines coming out next year from Europe that suggest that for most surgeries, we don't even need perioperative antibiotics, let alone postoperative antibiotics. When we looked at the conditions for which antibiotics were prescribed for, and these are just high level categories, we did drill down and get specific diseases, but you can see in dogs GI, almost 42% of cases of GI disease were prescribed an antibiotic. And I would say most of those cases are those acute diarrheas that don't need an antibiotic. And almost half of cats presenting with respiratory disease were prescribed an antibiotic. And cats often get upper respiratory infections in which they really don't need an antibiotic. Most of those resolve on their own. Most of those are viral in origin. So again, these are really nice areas to focus in on in terms of targeting antimicrobial stewardship efforts. And here, this is just a really high level look at disease categories and whether or not there's evidence of infection. And the thing to me that's really interesting is that for things like skin disease, which is like the skin is on the entire outside of the dog and really easy to sample, most of the time we're not even confirming infection. And that would be as simple as just taking a swab and looking at it under the microscope, which is a patient side point of care test that's cheap, easy, and we all learn to do it. Our technicians learn to do it. So there are a lot of missed opportunities. So when we look at adherence to guidelines, and I didn't share with you this data, these are just kind of summary things that were interesting from the data that we collected in these practices. 17% of sporadic lower urinary tract infections were treated with first-line agents. That means that most of them were not. 11% of routine spay-neuter surgeries received prophylactic antibiotics, and that's a clean elective surgery that shouldn't require any antimicrobials. 42% of dogs with GI disease received antibiotics, or I talked to you about that. 77% of cats with an upper respiratory infection received antibiotics in what we consider a watchful waiting period, so when they've only had clinical signs for less than 10 days. So the guideline recommendations are not to treat in those cases. And then when they were treated, whether or not it was appropriate in beyond the watchful waiting period or not, only 31% were treated with recommended, guideline recommended antimicrobials. 21% of dogs with superficial pyoderma, so skin infection, where we have the opportunity to treat topically, only 21% of those dogs were treated with topical antibiotics. 78% were treated with oral antibiotics. So treating the entire body, including the gut microbiome, which causes dysbiosis, but also selects for resistant organisms within that environment. So this leads us to lots of data targets. So improving utilization of topical therapies, encouraging use of first-line antibiotics for common infections like urinary tract infections, encouraging the use of alternative therapies for GI disease, identifying barriers for antimicrobial susceptibility testing. Only 10% of patients treated with an antibiotic had a bacterial culture and susceptibility. Encouraging use of cytology and UA. So let's look and see if we have bacteria before we apply the antibiotics. And then it also really suggests that we need more studies and guidance for the use of surgical antimicrobial prophylaxis. So just really briefly, one last study. I'm not sure on my time because we started late. So just tell me if you need to give me the hook. As I mentioned, we kind of took a deeper dive in a couple of different areas. So one was dental disease, and this is published if you're interested. Common condition in dogs and cats, or common reason for presenting to the veterinarian, which would be for dental cleaning or dental issues. In human healthcare, about 10% of outpatient prescribing is done by dentists, and that's probably true in vet med as well. But there are no prescribing guidelines in veterinary dentistry, and in fact, that's something that's happening as a result of some of this work. Those are in the works right now. And there's also variability among dentists in terms of what they think, in what situations they should apply antibiotics. So there's not a lot of consensus, and guidelines are lacking. So we had 22 clinics participate. 19 were non-academic referral hospitals and 3 were teaching hospitals across the US. We had them include just the first 10 patients that presented for a procedure. So not just kind of a check or, a visit prior to a procedure. We really wanted to focus on antimicrobials prescribed around procedures. So dental cleaning or surgical procedures or dental extractions. And we collected sort of all of the typical data about the patients, the clinical conditions, what procedures were performed, and then data about antimicrobials that were prescribed. So we had 222 patients. Again, most were dogs and most were outpatients. And, oh, that's not showing up. Emma, do you remember? I think it's like 35% of these patients presenting for procedures received an antimicrobial. And again, those beta-lactams that are potentiated were most common, and then clindamycin was the number 2 drug. And you see this cefavecin, this nasty little cefavecin, that 3rd generation injectable cephalosporin, which is like not an appropriate drug for oral therapy, showing up here too. So vets like to give it because it's easy. 48%, so of all of the antibiotics prescribed, almost half were for the purpose of prophylaxis. And again, when we look at duration of treatment for the purpose of prophylaxis versus when we're actually treating an infection, we can see that for dogs, 7 days was the median for postoperative prophylaxis, while 10 days was the median for infection treatment. And there are smaller numbers for cats, but the same trend is there too. So we're treating almost as long when we're trying to prevent infection as when we're actually treating an infection. We looked at risk factors for antimicrobial prescribing, again, to try to find areas where we might be able to intervene. Comorbidities, so other disease processes, periodontal disease alone, or the placement of implants in our study were not risk factors for antimicrobial use. That might not hold up if we repeated this or we did it on a larger scale. But what we did find was that if there was periodontal disease and tooth extractions, that was a risk factor for prescribing an antibiotic prophylactically, and the number of extractions influenced the propensity to prescribe. So in human healthcare, there's just not a lot of support for using antibiotics for patients that have teeth extractions. We know that dental prophylactic procedures can cause transient bacteremia, but it's not actually more than just brushing your teeth. So there's this perceived risk, which is probably not real. And antibiotic prophylaxis is rarely indicated for patients with implants, and that can be a reason why a lot of dentists, veterinary dentists, prescribe antibiotics, because they're worried, oh, they had a hip replacement or they have an implant in their knee. And really, there's just specific targeted patients in high-risk situations that should be getting an antibiotic in human healthcare, and those are things like previous endocarditis or cardiac implant. And so again here, because so much use in veterinary dentistry is for prophylaxis, one of the outcomes that we identified from this study is that we really need to determine in vet med when are prophylactic antibiotics necessary for veterinary dentistry. And again, there's a group working on some consensus guidelines, but unfortunately what we really need is more studies specifically asking this question. So in summary, we collect antimicrobial use data to establish baseline measures, identify areas for improved prescribing, identify areas the veterinarians just need more support for, monitor progress over time, and then monitor changing practices over time. So we can make progress in certain areas, but as new things come out, like injectable 3rd generation cephalosporins, that really changes the way veterinarians practice. And so we need to continue to do these sorts of measurements so we can capture those things as they occur. And I think that's it. This is my dog. That's it.
Questions for Dr. Granick?
Dr. Geddes.
Yeah, this is wonderful to hear. I was just wondering, so a couple of years ago, had one of my cats had a little polyp removed from its ears, and he got prescribed systemic antibiotics as well as local antibiotics, as well as systemic and opioid, and like there was a lot of drugs prescribed. I was like really perplexed by all of that, and so I was just wondering how much of the corporations buying veterinary practices and the pressure for profit, how much of that might be influencing some of this? I just want to hear your thoughts about that.
That's a good
I don't know if that was the case with my cat, but just
yeah.
thinking about that.
Can everyone hear the questions over Zoom? I don't have to repeat
Yep,
them.
they should be able to, yep.
Okay, so good question. I don't know that that's specifically been looked at. I do know, as Emma mentioned, we have this new network CAMS, Companion Animal Antimicrobial Stewards. And we do have industry involvement in that, including some of the large corporate practices. So I think that, you know, unfortunately, because the pharmacy is in the vet hospital, that can drive revenue. So there might be impetus to prescribe drugs just because that's a source of revenue. But there are efforts to try to improve prescribing, and that probably includes opioids as well, because that's another hot button issue in vet med. We want to control pain, but we don't want to overprescribe opioids. And so I'm hoping that we will make progress there, but there are definitely folks in corporate medicine that are very interested in moving the dial in terms of improving antimicrobial use, if that makes you feel better.
Great, thank you.
Susan.
This is great, thank you.
Yeah.
I learned of this study of all places from Checklist Manifesto by Atul Gawande, and I might be misremembering it, but in his book, in Checklist Manifesto, he describes a very large human study where for prophylactic use of, I don't know what it was using, I'm going to presume cefazolin, but something like that, for intraoperative use. What they found in this large study was that the administration of, I'm just going to say cefazolin, before the first incision was made, was observed to be protective against surgical site infections. Do you know if any similar studies have been conducted in veterinary medicine?
Yeah,
I don't know where we get get that from, but yeah.
So I believe there are studies in vet med that look at that and look at timing specifically, and that there's studies looking at, you know, like PK/PD and when you need to redose those. So it's kind of like within the hour, I think, of the first incision, and then within 2 hours of, you know, each dose if surgery is ongoing. And then the thought is that post-surgically, if there's a need for prophylaxis, really at the 24-hour point, there likely is no need beyond that because you have some sort of fibrin seal at that. But I do believe that there's some data in vet med. But don't quote me on that.
Just to follow, thank you. And just to follow up on that, do you think that the current, because our current, I would say, and maybe Alonzo, you can comment on this well, but I think our current default is to administer cefazolin here prophylactically.
Yes.
Do you think, is that something in the future we're going to be looking at, examining whether or not that's something that should be our default here? Or should we not be doing that as our default?
Yeah, I think there are surgical prophylaxis guidelines coming out from Enovet. They use a lot less antibiotics in general in Europe, so I don't know that the uptake of those guidelines, what they're going to look like here. I think there's going to be quite a bit of resistance.
Sure.
Unintended. Right. Yeah, unintended. But I think for implants, there's pretty good, especially for TPLOs, there's pretty good evidence that perisurgical prophylaxis is important. The question is whether we need drugs post-surgically.
Okay.
And one of the things that we've observed in the data that we've collected from our own teaching hospital is that cefazolin is the default for surgeries. And weirdly, for some surgeries where they go home with post-operative prophylaxis, they go from getting a perioperative, which I would think is the most critical period, first generation cephalosporin, to a post-operative third generation cephalosporin. And there's some question now, like one of the pharmacologists at OSU presented to us recently, that there's some question about the pharmacokinetics of an oral third generation cephalosporin, cefpodoxime, and whether or not it truly is efficacious only given once a day. And I think a lot of veterinarians choose that drug because it's a once a day versus a twice a day drug. So all of that stuff maybe will be blown out of the water soon. We'll see.
Okay, thank you.
All right.
I think in the interest of time.
Yes.
Thank you. Move on to our next speaker, Dr. Kathleen Boris-Lawrie.
You can borrow pictures of my dogs anytime.
It was very cute. It looked like it was in Arizona.
No, it was.
You're,
Yes,
It was just in
please go
southern
ahead and introduce our next speaker.
thank you, thank you. Good afternoon, everyone. It's a great pleasure that I get to introduce Dr. Kathleen Boris-Lawrie for today's talk. Dr. Boris-Lawrie, or Kathy as I know her, is a professor at the Veterinary Biomedical Science here at the U. She got her PhD from George Washington School of Medicine, and she's been focusing her research career on sort of the interplay between HIV infection and the host defense mechanism. She's done some good work on uncovering sort of the RNA epigenetic modification that happens in that kind of interplay, and she has sort of uncovered an area of host translation mechanism that not only important for HIV viral pathogenesis, but also mammalian sort of response to stress. So that is going to be a focus of today's talk. I have an honor of having her as a mentor and experience firsthand her sort of expertise and remarkable sort of characteristics, both as a teacher and as a researcher. So I'd like to have you join me in welcoming her up on stage.
I'm really pleased to be here, and I thank AK Hayir for the introduction. AK and I met when he was an undergraduate at McAllister, out at the U. And he phoned me up or whatever and asked if he could come and do some undergrad research because he was a biochemistry major over there. And he came. In one semester, he had such an impact on our research that he's part of a research paper with Dora Zucco, whose work I would like to mention today. And now he's working up for his own medical career, probably as a physician scientist, and working in our lab again. So thank you, AK. And I think I only have like about 2 minutes. Yeah, so you guys get the really good snapshot. And can you see that there's a play of words here? SOS, switching on survival. And this is about SOS translation, specialized translation, and what's special about it. And so I'm really not going to, I have slides and they were for 20 minutes, but even so, I'm going to make it really short. Let's see if we can do this in what, 5 minutes or 4?
I think you have plenty of time.
Just do it.
Oh, really? No.
It's recorded. Don't short everyone.
Well,
Yeah.
really, because people's schedules and things.
You're okay.
You're okay.
All right, well, I'll still be as brief as possible. So my outline is 3 parts. And one is, people talk about transcription all the time. What's post-transcription? Secondly, hypermethylation, what's that? That's when you epigenetically modify, not DNA, but RNA. And we don't do what other people generally do, which is look at the body of the RNA. We look at the cap, the beginning, like your cap, Adrian. And so it's got the little top on top, and I'll come back to that. And then I'll so we defined it by study of HIV. And people go, oh, why do you study HIV around here? Well, HIV is a great model. Retroviruses are great models. Bird retroviruses, bovine leukemia virus, for how cells work and for disease in general. Cancer, yeah. AIDS, immunodeficiency, sheep, Visna that's where it was discovered, immunodeficiency in retrovirus. And it can also, retroviruses can be vectors for gene therapy. So, yeah. And then really, this is where we are now, and AK is working on this, and that is the adaptive. I want you to be able to say at the end that what this is and what this is. And this is what RNAs are actually translated. Okay, so a lot of people do proteomics and transcriptomics. So people take their cancer cells and isolate the RNA and sequence it. And then they say they have various RNAs that are expressed, and those are cancer RNAs. So, but when you look at the proteins in those same cancer samples, they're really different. If they were the same, this peak would be like this. It's way pushed over. And that's because there's this discordance between what the RNAs are and that have the capacity to be made into protein and which are the actual proteins that are there. So why does it matter? Well, imagine if you're taking chemotherapy and it's against gene products of these RNAs, and those gene products aren't there. Ooh, it's not good, right? And so we want to help the field correct that kind of a thing. And that is just some specific genes like P53 and other important tumor suppressors. So now I just go back. This is my first section, and just to define some terms. We all know DNA, double-stranded. And I'm talking today about RNA, specifically the M, the stuff that's the messenger that's going to be translated by these ribosomes into proteins that fold. And new proteins fold, and steady state proteins fold, but these proteins experience life. They got hit by the UV rays. They got hit by the cisplatin or the antibiotics that we just took. And so they might hang around for a long time, or they might not. So if we're going to measure what is happening in our cancer cells, we really need to look, well, great to know everything, right? But we're concerned about these freshly made proteins and what they look like and what they are. And we really want to know, what are the proteins in there that we need to battle? And do we have the machinery for our chemotherapy to work? And I would say that we probably don't in some cases. So some terms here, and all the genes that get expressed are the transcripts, and the proteome is the proteins. And this is the term that's not so well known. It's the translatome. So it's the stuff that gets translated. So translation is the process by which RNA information gets turned into proteins. So DNA, RNA, proteins, the central dogma that Kevin teaches on in our microbiology course. Okay, so this is part 2. And so an RNA, when it's born, on that last slide, it was a blue thing and it had a ball in the end. That ball is, okay, so you have the RNA and it starts at a certain sequence. All RNAs get modified at the beginning. They all have their transcription start site, but that will be, the RNA will be like an unwound molecule that gets degraded unless it has something to protect it at the 5' end. And so if this is the RNA strand, it gets protected by another RNA, a ribonucleotide. This is a guanosine. And then, you have A, G, C, T, or U. And you get a backwards, so usually it's 5' to 3'. You get a backwards 5' to 5' guanosine molecule stuck on the end. So it's basically like this, and you get a backwards G. And so the enzymes that degrade nucleic acid all go 5' to 3'. But now the other end's sticking out, so it's harder to decay. And then proteins bind, and that's all really interesting to us because they determine what happens to that RNA. And that determines which ones get made into proteins or not. So that gets the backwards G and it gets a methyl group. And so AK showed this in red and this special linkage in red. And there's lots of different kinds of RNA. mRNA is just one kind. It's kind of like it takes a village. You need spliceosomes and ribosomal RNAs and all these different tRNAs, all these different kinds. And we all pay homage to the mRNAs because that's what is made into protein. Well, those other guys they get the same cap. So here's my cute Minnesota cap. You guys like this? I think it's cute. That it's a backwards G that's on the body of the RNA. And it got a single methyl group. So non-coding RNAs often get 3. And they're all the same kind, but they make the shape of the cap different. And that looks kind of mild right here. But actually, if you were walking around, Adrian, with 3 dots, people would be going, why are you having 3 dots on your hat, right? And so the enzymes in the cell actually also react very differently to this than to this. And so this just shows that this lucky nitrogen gets 2 extra CH3s. So why do we care? So here's a little structural biology, just one slide. And so lots of people have studied this, how it binds to proteins. And they even have crystal structures and now cryo-EM structures. And basically, the gray parts are the amino acids. And so the protein is kind of like this. And these W's, tryptophan, is that right? Tryptophan. Amino acids, they kind of like hook on to that guanosine planar molecule in blue. And it's really neat. You can actually see this. And these are both the same family of proteins. This does translation. And this is the enzyme that holds on and is going to add another methyl group to this. It's amazing. But when that happens, the space in there is too tight. And it gets other proteins help cast off this to bind. And instead, when it's trimethylated, it binds to different proteins. And those different proteins help it become a spliceosome, which does the RNA splicing that is so important for alternative splicing and making all kinds of different proteins. We can have only so many genes, but we have lots of RNAs because of alternative splicing. Okay, so that's really cool, all right, and that's because of that. But these are all non-coding RNAs. So in the field of retroviruses, it was shocking when a colleague of ours, T. Zhang, identified that HIV RNAs have this sometimes. And that happened in 2010. And he sent me the antibodies and asked me to repeat his experiment. And we couldn't do it at the time. And actually nobody else could do it either because it was really hard. And we went on our merry way. Well, then my lab was studying, he sent it to me because we were studying RNA and retrovirus. So we went on our merry way, and then we started to look at translation and some of the proteins that bind to this cap. We expected it to be this guy, but we found out it wasn't. It wasn't that there was a difference in the cap binding protein. And so that led us to look and see if, well, maybe T was right. And so now we could do this, partially because of the great people that I had in the lab at the time, including Gadi Singh, who's a professor now, and Brad Seifzer, who's in the lab today. And they were able to we were able to do the experiment. And this, I'm not going to go through data. I'm just going to say we made some pretty cool diagrams. We like diagrams. And we were able to take ribosomes that had a lot of RNA on them. That means they were very busy making proteins. And we pulled those out and we stripped off the RNA. And we looked to see what the cap was. And when we did that, what we found is that if we originally took these ribosomes and we fished out the RNA using a tag to that 4E, EIF4E, we did this and did PCR to amplify up the RNAs. We didn't find the HIV RNAs we expected, but we did find these multiply spliced ones. And they're in different families. And the cool thing is these RNAs get translated into the structural proteins that actually make the virion. And these are the early RNAs that tell the cell to make more of the virus. So I'm going to cut to the chase and just tell you, 10 minutes is up, that these guys have the normal cap and these have the trimethyl cap. And by studying this, do we say this up there? So they have 2 different ones. And by studying this, we've been able to group these RNAs with housekeeping RNAs that a lot of us use in our experiments, GAPDH, and we were able to group these with some cancer RNAs, like P53, the guardian of the genome. Okay, and so, we did other experiments and we looked for others, which if we can knock down the proteins, what happens? And I just want to point out right here that if we have cells, let's see, no, this is the wrong picture. If we have cells and we treat them with it, well, I'm just gonna really cut to the chase. Let's see here. Yeah, do you guys like this picture or the gels better?
Gels.
The gels?
No. No, this is me.
Okay, well, so in the clinic, rapamycin is an old-time drug that's used in some cases to inhibit protein synthesis. There are now small molecules that do the same job, and they're in the clinic for cancer and other diseases. They stop the RNAs that have the single puff cap. We discovered this other pathway. T discovered the TMGs, and then we put together what are the proteins, and they were nuclear proteins that bind to this cap and stay with it for life. And then it goes to the cytoplasm, and those nuclear proteins load up the ribosomes. And that was heretical to think, right, Kevin? Even as bacteria, they do it all in one compartment. But eukaryotes separate these compartments as another way to regulate things really carefully. So nuclear things are supposed to stay and do nuclear stuff. And then when you go to the cytoplasm, 4E is there, and you use those proteins to use translation. No, we found out that the nuclear proteins could then program the translation of select RNAs. So select RNAs are licensed by the trimethylguanosine cap, TMG. And what we discovered was that if you knock this out, the cells would slow down their proliferation. If you knock this out, cells would slow down their proliferation. If you knock this out, let's see. And if you knock this out, however, and you inhibit this, the cells die. So in other words, we've applied this to cancer cells and so Dora applied it to the osteosarcoma. She's was a graduate student. She's graduated now in the VMed program. That if we would inhibit osteosarcomas that we got from Jaime Modiano, which in the VCS department, it was very important interaction with him to study the osteosarcomas, that we would, if we knocked out the mTOR pathway and then we washed out the drugs, cells were happily growing in anchorage-independent growth assays, which is the state of the art for cancer studies in a lab. If we knocked out the TGS1 pathway, the cells wouldn't grow. If we knocked out, if we added the TGS or TMG pathway, we could kill the cells and make the cells not be able to recover from the mTOR inhibitor. In other words, I'm not showing you the data, so it's kind of hard for you to believe me on this, but in other words, people use mTOR inhibitors in the clinic and they wash out and then the cells start growing again. If we knock down this specialized translation pathway, they no longer can recover. So these pathways are like this. And if we could go into an animal model today and use our approach that we developed in the lab, we believe that we would be able to eliminate the cancer growth. And so those studies were with osteosarcoma cells, and we would love to be able to collaborate with someone who is interested in a mouse model, probably a mouse model first, and then move into a canine model to test what we found out in our assays. Okay. So here's data. It's really cool. Yeah. Okay. Here's our caps. And this is where, now I don't have to hand wave. I can just quickly tell you, there's 2 slides left. these are growth and low attachment plates. So cancer cells, they have hallmark features, one of which is anchorage independent growth. That just means that, and they can grow without growth factors. So we took these osteosarcoma cells and put them in that kind of environment. And they would grow. So that was measured by light units with a reporter. And if we treat them with the mTOR inhibitor, that doesn't have off-target effects, it's really specific. And then we wash it out and then look at their growth, they grow. But if we knock down our pathway with these, we use siRNA, usually, RNA interference, and knock down these proteins that are necessary for the specialized translation pathway, there's less growth. And if we add the mTOR inhibitor, there's even lesser growth. It diminishes more. And if we put these two together, so we can really, really reduce that. And those cells, well, they're not growing, they're dead. Okay. And we could do that in a couple different ways. And we could also treat with a CREM1 inhibitor. So leptomycin B is part of a class of protein that also has been made into more contemporary single molecule drugs that's also used in the clinic as an anti-cancer drug. And what we showed in our paper is it blocks, I told you about TGS1, it hooks up to the single cap and then it methylates it. Well, this drug sticks its nose in that active site and doesn't let TGS1 work. So we can block the specialized pathway using this drug. So this was another type of experiment. Two ways to do this experiment. We knock down both pathways and the cancer cells don't grow. So now we know this. And now what we're doing in our real last slide is to characterize what are the proteins that are being made on the specialized translation pathway. And the way we are doing that is, this is more data. We love this figure, Kevin. You can see very clearly when you add the mTOR inhibitor, if this is the amount of translation, it brings it way down. And so these little guys, these little bumps, are the amount of translation by our specialized pathway. If we knock down both, it'd be flat line. So we want to know what's being translated here. Okay. And so we're doing that by just, these are physically the ribosomes attached to RNAs. And so we just now isolate these complexes and isolate the RNAs and use RNA-seq analysis. So we identify the RNAs that were actively being translated. So that's pretty cool. But we want an insurance policy. So with Stephanie Thomas in the medical school, who is a proteomics expert, we take these same complexes, and you can imagine those ribosomes making the proteins. The proteins are just coming off in real time. And we can lock them on, give it to Stephanie's lab, and they can isolate the proteins, the peptides that are associated, and do mass spec. So then we know the protein sequences and the RNA sequences that are actually being translated at that point in time. And so what we're doing, let's see, is we're looking for candidates, and we're also looking just to discover. But we want to look for candidates so that we can go back and test and validate. And so I think, oh darn, I didn't put it. So what we're doing is we're looking in the presence and absence of chemotherapy. I lost my slide. So we're looking with chemotherapy and without chemotherapy. And we're looking to see what the differences are. And so one of the drugs we're testing is a PARP inhibitor. So PARP inhibitors, polymerase. Yes, it helps things get DNA damage degradation. They get recognized, DNA damage gets recognized, and then that's a signal to repair it. So people so ovarian cancer, for example, human ovarian cancer, people are treated with PARP inhibitor. So that then they are given cisplatin or some other DNA damage chemo. And that's supposed to make it so DNA damage happens and the cell cannot repair it, so it'll die. So what we found in our studies, targeted studies, that PARP1 isn't even being made into a protein. So that means that in the model that we looked in, which might not be like patients, these are patient tumor explants, things could have happened and changed, but there's no reason to give these cells a PARP inhibitor. So what should we be giving them? That's our question. And that's what we're trying to identify. Okay. So that's really the end of my talk, and I did it in about 20 minutes still. Okay, so thank you for letting me speak. And I want to point out, Dora, graduate student, did a great job. But we're ready to do animal studies, so please talk to us if you're interested. AK, Brad, and others. Jaime, Stephanie. So thank you for your attention, and any questions, I'll be thrilled to answer. Sure, people need to move on. Wait. There's Jen's dog again.
Oh, that's good.
Rob, yeah.
Okay, so for the PARP inhibitor, and you say you're looking for other targets, right? Do you expect those targets to be in that DNA repair? Right, because they were looking basically at something where they destroyed the DNA and then the cell can't repair it. So are you expecting
Yes.
targets?
Yes, we're looking at, like, for example, BRCA1 is on our list there. So yes, we're looking, yeah, we're developing hypotheses to dig into certain theories. And so that is one of them. Yes, and so we would like to know what are the other DNA damage repair proteins that are being translated into protein and could be functional. And so let's say BRCA1, well, ovarian cancer, that's often, the ones we're using are BRCA1 mutated. And so you would think that DNA repair that wouldn't work. And so we'll see if it, so when we sequence RNA, if we use nanopore sequencing, the second generation sequencing, we can get the isoform of the transcript that's being made. And so I mentioned post-transcriptional control is about after transcription, you get the processing of the RNA and the cap. You also get alternative splicing. So we've been able to pull out different isoforms. And so we would like to know if BRCA1, so there might be a different isoform that could be used. Yeah, it's really, there's a lot of different elegant sort of molecular questions you can ask and then go back in and look at the proteins and the protein activity. Yeah.
That was great, thank you. It's a very elementary question, but
Great.
we're thinking about the population density of the transcriptome and specifically, that which is destined to become protein. What's the density of the transcriptome relative to the translatome? Is there work to discover, to determine how many of those mRNAs actually do go on to become, to undergo translation into protein?
Yeah, that's a great question. And that gets to what is the bottleneck between the transcriptome and the translatome. I don't have a number for you, but I can tell you that when we're looking at what proteins are being made, we're looking at the proteins that do the RNA processing and that do the nuclear export, and then the canonical translation pathway. So we're looking at to see if the machinery is there for the RNAs that get made into proteins. And so one of the things in our big picture of what we discovered is that there are cohorts of mRNAs that use different translation pathways. So we would like, and so we have the mTOR inhibited one. That's those are usually RNAs for proliferation and transcription factors that load up with that translation machinery. So in cancer, generally, translation is dysfunctional, and that pathway could be hyperactive or inactive. And so that's the way that we can start to look to see, you know, really think about which ones get loaded into the polysomes.
Thank you.
Yeah.
All right. Well, I think
Okay,
in the
thank
interest of
you.
time, thanks for sticking with us.
Yeah, thank
And
you.
let's think. Thank you so much. It's great. And we have a gift
Oh,
from the research office. This one is for you.
thank you.
Thank you for being a part of our seminar.
Oh, thank you. This is cute too.
I know.
That's what it's there
Thank
for.
you, research office.
Maybe I need people who have one of those.
Yeah.
It's like, I learned so much in this.
Okay.
Yeah.
I've never actually seen it, but that's awesome.